I have very great admiration and respect for the work of the Academy, and so I was delighted to be asked to take part in this meeting. I think the initial reaction of most people would be that regenerative medicine is very high-tech, and so the answer to my question is no, there isn't, certainly at present, a role for regenerative medicine. What I want to show you is that some of the approaches are actually a little bit different and may be amenable to use anywhere in the world by the time we've finally developed them in perhaps 5 or 10 years' time. Generally, people assume that regenerative medicine involves cell therapy, the last of my three. I'm not going to talk about that at all. It undoubtedly will come along and be very important, but as I'll explain in a few minutes, it's actually very challenging. The other two routes are to identify drugs which can reduce the harmful effect of a disease, or to promote the body's own cells to repair the tissue. We happen to have examples of both of these in the centre here in Edinburgh. I'm going to describe these for you. But first of all, I think it's important to discuss what we mean by stem cells. What is a stem cell? Well, a stem cell has the ability to either form more cells like itself, which can go back into the population to maintain the population, or to produce other cell types. And this is why they have this unique and critical ability to form tissues during development or maintain them during adult life. If we're going to use them for therapy, we may use them either for research, in which case the requirements are rather less stringent. We have to have cells of the correct tissue type with minimal contamination by cells of the other type. And that's really it. If we're going to use cell therapy, not only do they have to be at a clinical grade, but they have to be immunologically matched to the patient. In comparative terms, you need a very large number of cells, and you also need to know how to put them and where to put them into the patient. None of these steps is trivial. And this is the reason why we would think at present that the use of cells for research is much more likely to produce a therapy than the target of having cells for use in transplantation. There are a number of different sources of stem cells, which I'm going to just describe quickly, and we'll see that none of them are perfect. There are limitations to all of them. The first populations to be identified were taken from tissue, and they're usually known as adult stem cells. And over a period of time, cells have been identified from most different tissues, including the brain and the heart, where they are relatively rare and divide very, very slowly indeed, but they are there. They have the limitations that, in general terms, they have a shorter life in culture and will only form the tissues from which they were taken. They may go slightly wider than that, but the comparison in both of these cases is with the embryo-derived stem cell, which are markedly different. The embryo-derived stem cells have two unique characteristics: that if they're well looked after, they will divide for a very long time. Therefore, you can have very large numbers of them. And they will retain the ability to form all of the different tissues of an adult. That's the criterion for giving something the name of an embryo stem cell, a pluripotent stem cell. This, of course, is what makes them so useful from the point of view of research and potentially therapy. But the drawback that they have is that if you put them into a person, they have the ability to form tumours, teratomas. And it's this risk, which I suspect is one of the important facts slowing down the move of stem cell derivatives, embryo stem cell derivatives, into the clinic. Most of you will be aware, I'm sure, of the trial in the United States by the Geron Corporation using nerve cells derived from embryo stem cells. It's known that they put 22,000 pages of data into the FDA. 22,000 pages. I don't know, but I guess that a large part of that was showing that they weren't inadvertently putting in any remaining embryo stem cells. The other part, of course, will be showing that the effect was efficacious in the animals that they studied. I have a particular pride, if you like, in the fact that by far the main inheritance of the Dolly experiment was to make people think differently about development. Just to remind ourselves that all of our different tissues came from the single cell embryo, which is about 1/10 of a millimeter in diameter. Almost all the cells in our body have the same genetic information. And for many years, people wondered, how did that single cell give rise to all of the different tissues? We now know, of course, that there are genetic instructions encoded in the chromosomes which direct that differentiation. There was then a period of time when people thought that we couldn't change cells from one type to another in a tissue culture dish because the mechanisms which directed them to become that type of cell were very complex and rigidly fixed. So once you had a skin cell, a muscle cell, a nerve cell, terminally differentiated, as the name implies, that was as far as things could go, and you couldn't make them go back. So the effect of Dolly was to make people realize that under some circumstances that's not true. That in that case, magic factors in the egg had got the ability to influence the genetic information in the cell, which came from mammary glands, if I can remind you, so that it had the ability to go all the way back to the beginning of development and then support normal development to term. So it made lots of biologists ask the question, well, if the egg can do it, are there any other ways of taking a nucleus, a cell, to an earlier stage or perhaps a different stage? And the first person to achieve that objective was Japanese scientist shown here, Shinya Yamanaka, who did a very systematic experiment taking transcription factors which were known to be either essential for embryo stem cell function or at least for them to be present in embryo stem cells and introduce them into skin cells. He initially started, I think, with 22. Transcription factors are the proteins in cells produced by one gene which influence the function of another gene. So they, between them, are responsible for determining the nature of cells. And what he found in the end was that the list of just 4 genes here was sufficient to change a small proportion of skin cells to be very, very similar indeed to embryo stem cells. Embryo stem cells are called pluripotent, meaning they have the ability to form all tissues. And so he called them induced pluripotent cells. And the jargon phrase, of course, is then IPS cells. I'm quite clear in my own mind that that experiment was one of the most important of this decade, that decade, because it was before 2010. It may well prove to be one of the most important of this century because of the changes which it's introducing into biomedical research and ultimately therapy. So what about induced pluripotent cells? Well, like embryo stem cells, they have a very long life in culture. They retain the ability to form all of the tissues. They're essentially indistinguishable from embryo stem cells. That also means, of course, that they have the disadvantage that if you inadvertently transferred any into a patient, there would be a risk of a tumour formation. The one difference that there is from embryo stem cells is that you know the genetics, which is a jargon phrase, just to explain why that's important. If you were wanting to build a library of cells for transplantation, you would need to choose them to be of a particular immunotype. With embryo stem cells, you can't really do that because you can't know the genotype in advance. With IPS cells, you can. And there's no doubt that different groups in the world now are beginning to go looking for people who are homozygous at the key antigens. Homozygous meaning just that the 2 genes that they have, the 2 copies of the gene that they have are identical. Because this means that their cells are much more useful for transplantation into other people. Once the principle of changing cells, jargon is reprogramming them, was accepted, people began to ask, well, okay, if we can go from one tissue type to pluripotent, can we go from one tissue type to a different tissue? And the answer is yes. By identifying key transcription factors for the tissue that you want to produce and introducing them into skin, it is possible to produce at least the tissues that are shown here. The initial studies produced terminally differentiated cells because most was known about the transcription factors in those cells. But there may well be important reasons for producing progenitor cells. Now, progenitor cells are somewhere between the pluripotent cell, the adult stem cell, terminally differentiated cell. They're somewhere in the middle here. They do have some ability to multiply, some ability to form different tissues, but they will not form a teratoma if you were to transplant them into people. There may be problems in other ways in that they may not integrate normally into the tissue that you're trying to repair. So we mustn't be too naive in our ambitions, but there may be benefits in producing progenitors from the point of view of therapy. So in summarizing this part of the talk, I personally believe that the ability to change the fate of cells will become routine. The procedures will be simplified and the accuracy of the change will be improved. Production of iPS cells is pretty accurate now, but production of the different progenitors is still not. Further progress is needed there. So this means that we have sources of cells for research, to establish a library of cells for cell therapy, and there are estimates made that about 150 carefully selected lines would provide a good match to everybody in this population. A slightly different 150 would provide a match to, let's say, the Japanese populations, Chinese populations, where the pattern of genes is rather different. So you can imagine a library of iPS cells or neuroprogenitor cells, whatever it is, of 150 different types, and then they would be available relatively quickly for use in therapy, just by thawing them and multiplying them a little bit. There may also be opportunity with cells like this to change a gene before you put the cells into, perhaps, but most likely a child. So if that's correct, if I am correct, I think people will look back and feel that this is the beginning of a new era in regenerative medicine. So it's usually expected that regenerative medicine will involve cell therapy, but as I've already mentioned, I think there's a very strong case that they'll be used first to identify drugs. So let me give you just two examples. There are many inherited diseases for which there is no treatment. So the idea would be to take tissue from the person to produce iPS cells to produce the tissue which is affected, and to be able to compare those disease cells with healthy cells of the same type. For the first time, to be able to try to understand the mechanisms which cause the abnormal functioning of the cell. It would then be possible to use the cells and the understanding of the cells to set up high throughput assays, looking for small molecules which would be able to reduce the effect of whatever was abnormal in those cells. Obviously, the drugs would have to be tested in animals before they could be used in patients. This will only work with inherited diseases, and so the hope has to be that the mechanisms in some sporadic cases will be the same as those in inherited cases, in which case the medicine will apply, but it may not always be the case. In motor neuron disease, for example, it's a family of different diseases. The first gene that was associated with the disease is SOD1, and it appears to act through a different route from the others. So if you were following that route down, it would look as if the treatment that you would develop with SOD1 would not be widely effective. The gene which is involved in the other cases, in more than 90% of cases, is called TDP43. TDP43 is a processing gene present in all cells, but the abnormality only seems to have a harmful effect in motor neurons and other nerve cells. So we have a project at the centre to produce iPS cells from patients with mutations in this gene, to examine them during differentiation, to look, because the abnormality which is seen in post-mortem tissue from patients is protein accumulation. We will look for accumulation in the cells in culture and look and see whether there's an effect on the life of cells in a tissue culture dish. This is not nerves, it's actually the iPS cells themselves. But when people looked at the lysate in these cells, there's rather more, this is just the acronym for the mutation, there's rather more in cells from the patient with the mutation than there is in the control, both in terms of the soluble fraction and the insoluble, but particularly in the insoluble fraction. So we're even seeing the abnormal effect in the pluripotent cells. My colleagues then went on to look at the survival of the cells in culture, and you can see again a significant difference in the survival of the cells during culture compared to the controls. So we're obviously very excited by this and very pleased by this, because it does seem as if we're making significant progress. But it's very important to point out the limitations. That so far we've only looked at the nerve. There's often in this disease there's an effect of the neighbouring cells, the glia, and so it's we're partway through the process of differentiating them and studying them. And I've only mentioned one case, so it's extremely important that this is repeated. Now it happens that there's a group in Japan who've looked at a number of cases with mutations in this gene and who similar things. So it looks as if our observations are being repeated by other people, which is very encouraging. So iPS cells offer us a huge advantage because we can produce the different tissues. You actually don't need to know the causative mutation as long as you're confident that it is an inherited case. If you've got a disease which has passed through several generations, it may well be inherited, and then you could use this approach even without knowing the mutation. Not all the mutations cause the disease in every person that has it. And what's believed to be the case there is that there must be differences in other genes which are able to protect the person from the abnormality. And this approach gives us a means of looking for the mechanism of these effects, in that you could have several cell lines from the same family, some of which had had the disease and some not, and then you could look for the gene that was present when the disease did not occur. There are a huge number of inherited diseases, hundreds, but there are also inherited cases in among the common diseases like Parkinson's disease, even motor neuron disease, which we've chosen to work on. Inherited cases are only about 2 or 3% of the total population. 97% it's sporadic. But this may well give us an insight into the mechanisms causing the disease. I'd like to move on and quickly describe a different approach being led by the current director, Professor Charles French Constant, whose interest is in multiple sclerosis, which reflects the failure of nerve cells to repair the myelin, which is the insulating sheath around the neurons. Now, demyelination probably occurs in all of us. The disease occurs when we haven't the ability to put it back. Our cells haven't the ability to put it back. So what they've done is to study gene expression in tissue where it is going through a process of repair. So what this is showing is a plot of different genes. In some cases a reduction in expression, in other cases an increase in expression. And they had several plots, I'm only showing you one, chosen as being the time when remyelination was taking place. And they picked out that several genes were all part of one system, and in particular this very high level of expression was a gene, retinoic acid receptor, one of the particular genes of that family. And so they've done other studies looking at the level of transcripts in sclerotic lesions, again from post-mortem cases. And in humans, there is again this repeated presence of retinoic acid. They did other experiments deleting the gene in animals and showing that it did not, the animals were not able to remyelate the nerves. The most interesting thing of all was that they found that if they prompted gene activation in an animal model of the disease, it improved remyelination. So you can see the line of thought is that this is suggesting a molecular mechanism for promoting repair. And there's no doubt that within a few months they'll be looking for small molecules which can do this within animals and ultimately within patients. So what I hope I've shown you is that there are two ways of promoting recovery, of providing regenerative medicine, shall we say, which don't demand particular high tech from the point of view of delivery, and which may be amenable for use anywhere in the world. Thank you very much.